or producing most of the structural elements within and around cells. So enzymes in general, or enzymes specifically and proteins in general, are hugely important to both the structure and the function of a given cell. So if a certain cell is transcribing only certain genes and not others, that's going to dictate not only what it's made of and how it's built structurally, but what it can do functionally, what it can do biochemically. So for example, a cortex cell in a root and a mesophyll cell in a leaf are different both structurally and functionally because they're expressing different genes. Different genes are turned on and other ones are turned off in these two different kinds of cells. Okay, so that's sort of a basic explanation of gene expression or differential gene expression. This is very, very basic, right? If you've taken genetics, even just genetics, if you're taking more advanced classes, this is very elementary. But just sort of a basic way that happens. Here's a simple model of a eukaryotic gene. You've got the amino acid coding sequence, all right, and so you're going to have A's, T's, C's, and G's in there, and those are going to be translated into messenger RNA and then translated one codon at a time, right, to give you a particular amino acid sequence. Well, it's in this part of the gene that the amino acid coding sequence is found, but sort of in front of that thing is a regulatory sequence. It's going to border the coding sequence, it's pretty close to it, and that regulatory sequence is part of what determines when the coding sequence will be expressed, or when that coding sequence will be transcribed into a messenger RNA. The regulatory sequence itself, however, does not have any, there's no protein code there. It's really a switch, all right, to turn that coding region on or off, or to determine when that coding region will be transcribed and then translated into a protein. Okay, so I think that's a review for a lot of you. Now, there is a class of proteins called regulatory transcription factors that will bind directly to the regulatory sequence of eukaryotic genes. And so that transcription factor binds to the regulatory sequence, and that either activates or represses sometimes, can turn that gene on or off. All right, either activating it or inhibiting it from being transcribed. So, for example, if a gene is activated, if the right transcription factor is bound to that regulatory sequence, RNA polymerase activity will be stimulated. That DNA will be transcribed into a messenger RNA. The messenger RNA will be transported to the ribosome, or will be translated into a protein. All right? Many of you guys have heard this before. When a gene is repressed, if the right transcription factor is bound to the regulatory sequence, RNA polymerase activity will be inhibited, and none of this will occur. Okay, so I'm not a biochemist, I'm not a geneticist, so I think about this simply. So I think about it sort of like a light switch, and maybe this regulatory sequence is like a light switch, and this transcription factor is like a finger, right? So I could go over there with my finger and turn that light on, or I can turn it off. Now, it turns out that you kind of need a different finger to turn the light on versus off, because there's a different transcription factor for each one of those jobs. But it's kind of like that. We're turning genes on and off with little regulatory transcription fingers. I'm sure geneticists would choke on that, but that's my perspective, I guess. It's a good analogy, I think. So here's a picture of that. Here's that coding region of the gene. Here's the regulatory sequence. Here's a transcription factor binding to it. All right, and in this case, it's activating it. So when the transcription factor is bound, then RNA polymerase transcribes that gene. Messenger RNA sent out to the ribosome, where it's translated into a new polypeptide, right? And that protein can be an enzyme. It can be a structural protein. It can be a protein that builds some other structural elements to serve as, the bricks and mortar of the cell. Okay, so what do hormones have to do with that, right? Because that's really our topic. Here's the deal. Many plant hormones are basically activating the activators, right? Many plant hormones are interacting with the proteins that then act as the regulatory transcription factors. So for example, we talked about this when we were dealing with gibberellic acid. We said that when you treat allurone tissues in that little wheat kernel with gibberellic acid, that there's an increase in the production of alpha amylase protein. All right. Well, that's because gibberellic acid plus a second protein are acting as a regulatory transcription factor that activates the gene coding for alpha amylase. All right. Gibberellic acid is binding to a different substance, a different molecule, a protein, and together they're going to the alpha amylase gene and they're binding to the regulatory sequence and turning that gene on. All right? If you treat allurone tissues with abscisic acid, however, I didn't talk about this, but if you treat allurone tissues with abscisic acid, alpha amylase production is inhibited. Well, that's because abscisic acid and a different protein bound together are behaving as a regulatory transcription factor that represses the alpha amylase gene. So here's an example where hormones are very directly behaving as regulatory transcription factors. Now they're bound to a protein, and they're kind of going and doing that together. But this is a very direct influence that hormones are having on gene expression. We're going to see in just a second that the hormone's influence is not always as direct. Sometimes there's a huge biochemical pathway between the original hormone message and the final action, but this is a very direct influence. Okay, and so just to step back for a second, the reason why plant hormones are so good as the reason why they're able to operate as chemical signals between cells is because they're working hand in hand with these target cells. So like I've said before, the original hormone message is important, but the target cells are just as important. And so here, we're not going to talk about this a whole lot, but here's a list of characteristics that target cells have to have in order to be good communicators with these hormones. These target cells have to be able to identify a specific hormone. They have to be able to tell one hormone from the others. They have to be able to measure how much hormone is present, right? Because a different amount of hormone means a different thing. They have to be able to transfer that information by way of a biochemical pathway. We're going to spend a lot of time on that step. We're going to see the original hormone message and then boom, what flows from that. And they have to be able to then convert that information into a complex set of developmental changes. So this is just a general list that makes a nod towards the target cells and acknowledging that it's not just the hormone message that's important, it's the target cells that are important. They have to be able to do all of these things in order to receive that message, right, and to do something about it. So they're working hand in hand. All right, now, one of the ways, building on that list then, one of the ways, or the primary way that cells recognize hormones are by way of proteins called hormone receptors. And many times these hormone receptors are found on the cell membrane. So hormone receptors. And each hormone receptor contains a very particular hormone binding site that's specific to a particular hormone. And when the hormone binds to the receptor, it activates a particular response pathway in the cell. So, for example, in this very simple diagram, I know these are shapes, but molecules have very particular three-dimensional shapes. So this isn't as silly as it looks. I know it's like boxes and wedges and arrows, but molecules are very dependent on their three-dimensional shape and fitting together in certain ways. So it's not that ridiculous of a picture. Here's the cell membrane. All right, here's the hormone receptor, and here's the hormone, and it's going to fit right in there like a hand in a glove. All right, and that's going to activate this receptor, which is then going to lead to a whole host of other things. All right, depending on the particular pathway, this might be a short chain of events, but it could be a really huge cascade of events as well. But this is the first step. Wow, isn't this screen just so crisp? Compared to the screen in our room, it's like, wow, like seeing these words for like the very first time.
Nothing's cycling through yet. Like that lube usually does.
Oh yeah, yeah. Well, I could probably make that happen too. So many times what happens is that the binding of the hormone to the receptor actually changes the shape of that receptor protein. All right? Which allows that membrane-bound receptor to then interact with other cellular components. So if we look here in this sort of simplistic example, hormone binds to the receptor, and look, the shape of the receptor changes. You can see here the shape of the sides have changed. But now all of a sudden, that receptor can now interact with these other cellular elements, here also bound to the membrane, because they now fit together like a hand in a glove or a lock and a key. Right, so hormone causes the receptor to change its conformation, we would say, right, to change its shape, which now allows it to interact with other cellular components. I've already told you then that, or I haven't told you this, but sometimes it's the receptor itself, that activated receptor itself may become the transcription factor. Like directly, that kind of goes, the example that I gave you with alpha amylase and gibberellic acid. That's sort of what I'm saying here. So sometimes this receptor then goes and, look, here's the receptor and the hormone, right? Behaving as a transcription factor to either turn on or repress this gene directly. Okay, so that's sort of the short version. That's sort of the very direct effect that hormones are having. Okay, so this is just sort of a molecular explanation for that alpha amylase gibberellic acid example in grass seeds. All right, it just gives you a little picture of that. All right, and in this case, if this were the case here, this picture is a little bit misleading because the hormone receptor in this case probably wouldn't be bound to the plasma membrane. All right, if the hormone and the receptor are going to travel down to the level of the gene and behave as a transcription factor, this receptor is probably not bound to the plasma membrane. Okay, so this picture, we might just kind of get rid of all of that. But as this picture shows, many hormone receptors are found in the plasma membrane. So let's look at this little pathway here. This is a little bit of a longer pathway. All right, pathway number 2. So hormone receptor is bound in the plasma membrane. Hormone binds to the receptor. Receptor changes its shape, right? And now the receptor can interact with these other membrane-bound components. And look what that thing goes into. Now this little B arrow thing is activated, and it goes and plugs into this little Pac-Man, right? This other membrane-bound component. And look what that thing does. It basically opens up a hydrogen ion pump. It activates a proton pump. Those protons now flood out in between the membrane and the cell wall, and it actually loosens up the cell wall. And we're going to see that that's really important later on in terms of different shape factors and morphogenesis factors. So We'll come back to that proton pump, needing the cell wall loosening a little bit later. So let's see how I've said it here. Hormone binds the receptor. Receptor